So we're going to talk about the very important principle of membrane transport. I want you to first notice that we have the cell membrane shown here in this cartoon with the lipid bilayer. And I just have shown here these membrane phospholipids with the polar head group as these little circles and the lines representing the hydrophobic tails. I don't have cholesterol shown here, which is another important component of the cell membrane, but for right now, this is how we're going to depict it. And the reason why we want to show this is that when we have molecules that need to cross the cell membrane, and these would be things like sodium, which is written as Na+. We have potassium, which is K+. We have bicarbonate, which is HCO3-. We also have calcium, which is divalent cations, 2+. We have magnesium, another divalent cation, 2+, chloride, minus, and even hydrogen, H+. Now, all of these are examples here of ions. And we also have ions on the inside as well. I'm not showing that in this example, but just kind of showing you that there's ions on either side of the membrane. There's also, just focusing on the outside of the cell, things like glucose and other relatively small uncharged molecules. We have, of course, water and urea and things like this. And again, I'm just focusing on the outside of the cell for now, ignoring that there's certainly things on the inside. And the idea is that these molecules I've shown here cannot get across the membrane easily by themselves. For example, we have our ions, which are charged. The hydrophobic tail, the interior part of the membrane, is going to restrict charged things from going through. So we need mechanisms to get these molecules across. And what we're going to focus on is two important classes of proteins that are going to help us get these molecules across. Let's start first with carrier proteins, okay? And what we can start thinking about with carrier proteins is carriers are really transporters. We often use the word transporters for these. And what these do, they bind the solute, the specific solute, and what that results in is a conformational change in the protein. And what happens then is the solute then is transferred across the membrane. Okay, and that is the idea, is that there's a binding step, a conformational change in the protein, and then that change is going to allow that solute to get to the other side. Now, there's a couple of mechanisms of how this transport could work. Let's start with first passive transport. Okay, passive transport. And what passive transport is referring to is it does not require energy input. So it does not require input of energy. And really the molecule moves from a high concentration on one side of the membrane to a low concentration. And we call that the concentration gradient, okay? In addition, when we're talking about charged molecules, and we're talking about these ions like sodium and potassium and bicarbonate and all those examples that we were discussing, there's also a consideration of the electrochemical gradient. The charge is going to have an effect too, and we'll talk about that later. Okay, so what we call this passive transport, because we're going from a high concentration to a low concentration, is keeping it simple. Of course, there's an electrical component in some cases. We can call this a facilitated diffusion. So you know how things move from a high concentration to a low concentration. This protein is just facilitating that process. Active transport is different. We sometimes call these transporters pumps. And the idea is that you are transporting a molecule against its gradient. It's going uphill, right? So we call this uphill transport, as opposed to the previous example, passive transport, which things were going downhill. So there are actually two types of active transport that we can think about. There's primary active transport, which you're transporting the molecule against its gradient. Again, that's uphill. And this would be coupled to energy input, for example, coupled to ATP hydrolysis, right? So this is energy input, right? Because if you're going uphill, that requires energy to do that. Now, secondary active transport, you are transporting the molecule using another gradient. And that other gradient was requiring input of energy. So you're using another gradient that was generated using, for example, let's say ATP hydrolysis as our primary energy input. So the idea is that for secondary, the molecule is taking advantage of the fact that another molecule already has a favorable gradient that was set up through the input of energy, and it's sort of exploiting that gradient. Now, let's talk about some examples of how this might look. And so what we can do is we can think about what that would look like. So here we are, we have our examples. And the first one is a very important one because it's on virtually every cell. And that would be using an example of the sodium potassium ATPase transporter, or sometimes referred to the sodium potassium pump. What this does is it pumps 3 sodium out, and in exchange for 2 potassium in. This would be an example. Again, this is the sodium potassium ATPase pump. Sometimes it's called the sodium potassium ATPase. And the reason why this is active transport is that you have a high sodium concentration on the outside relative to the inside, and you have a high potassium concentration on the inside relative to the outside. You see how potassium is going against this gradient as well as sodium. And this pump is very important because it maintains these gradients that we'll talk about being so important later. We also have examples where we have calcium getting pumped out, and the calcium goes to the outside, and again, it requires ATP hydrolysis. This is what we would call the calcium ATPase. So you see how that would be a calcium going by itself, okay? And then we have a couple of other examples I can show you. This also is a very important one where we have sodium going into the cell and glucose also going in. And the idea is that the glucose is going into the cell, and that's occurring from the sodium going down its gradient, which that was already created. Remember, the sodium is higher on the outside than the inside because of the sodium potassium ATPase maintaining that gradient. So that requires input of energy. The sodium's going down its gradient, and then glucose also can go down into the cell, taking advantage of that. And this transporter is called the sodium glucose cotransporter, or SGLT. This one's very important, especially in the kidney, where it's target of a very important pharmacological agent that is used in the treatment of diabetes mellitus. Another example, finally, would be another sodium example, sodium going in down its gradient that was set up by the sodium potassium pump, and then a proton going out, okay? And this would be an example of a sodium hydrogen ion, what we call an exchanger. You see how we're exchanging sodium for protons, and then this is abbreviated NHE. So I want to point out that in addition to these examples, we can also put some other descriptions on here. So you notice that for the sodium potassium ATPase example, the sodium and potassium are going in opposite direction, right? So this would be an example of what we would call antiport. One ion, in this case, is going one direction, and another ion is going in another direction. Another example of an antiport mechanism would be this exchanger right here, the NHE, because we have a sodium going one direction and a proton going another. You notice that for this calcium example, this would be an example of uniport. We have one molecule going by itself. And then you also have the idea here of what we call symport. That means you have, in this case, sodium and glucose going in the same direction, okay? So these are just terms that you'll come across, and a lot of these terms are used to describe these transporters, and it's good to have in your vocabulary. Next, we can talk about the regulation of how these different proteins function. And I'm not going to go through all the possibilities here, but I'll just give you a couple of examples. One possibility is you can have phosphorylation of the protein or dephosphorylation, and that could change the activity and even expression of the protein. So there are examples that we'll learn later that there's signaling from certain hormones, for example, that can affect protein kinases that can then regulate how these proteins are going to be functioning. And that's something that we'll discuss later. Another example is translocation sequestration. And the idea here is that some of these transporters can shuttle between being in vesicles where they're sequestered inside the cell. And when the appropriate signal comes across, those transporters can then be what we call translocated to the cell membrane. So you could imagine something like this, where you have the transport protein in a vesicle, and then there's the membrane, and these transporters can be inserted into the membrane, and they also can be removed from the membrane. And that can certainly have effects on the function of these transporters, because if they're not in the membrane, they're not going to work.